Hello everyone, welcome back to Swati's Bioscience Hub. So in last few sessions we have been discussing about the cellular respiration in aerobic conditions, okay? And we have talked about the glycolysis, the first step of cellular respiration. So in glycolysis what happens? In glycolysis glucose molecules break down to produce pyruvate, and along with pyruvate it also generates NADH and ATP. Okay. So what happens in this process? You utilizes the NAD plus molecule to produce the NADH. Okay. And this reaction takes place in the cytosol. So here what happens, this NAD, when the NADH is generated, this NADH has to move, this NAD plus supply of NAD plus is required to continue the process of glycolysis. Okay, NAD plus is required continuously for the process to goes on. So what happens, this NAD plus again regenerates from the NADH. Okay, and for that the NADH has to move to electron transport chain. Okay, to again produce the NAD plus, NADH must move to the electron transport chain. Now the electron transport chain takes place in the mitochondrial matrix. It takes place in the mitochondrial matrix. So NADH, the glycolysis process taking place in the cytosol of the cell, and NADH is getting produced in the cytosol of the cell, but it has to move to the mitochondrial matrix to the electron transport chain to again regenerate this NAD plus, and also in generating or synthesizing the ATP molecules. Okay. So, but what is the problem here? Here what happens, the inner membrane of mitochondria is impermeable to the NADH. Okay, inner membrane of mitochondria, NADH cannot directly penetrate into the inner membrane of mitochondria. So what happens? Here, this NADH has to enter to the mitochondria in some, in some other forms or through a different kind of system. Okay, and these systems are known as shuttle systems. Okay, so today we are going to discuss about this type of shuttle systems. There are mostly mainly two types of shuttle systems. So today we are going to discuss these two shuttle systems. So there are two types of shuttle system. First is glycerol 3-phosphate shuttle system, and second one is malate aspartate shuttle. Okay, there are two main shuttle systems that helps in transporting the NADH molecule to the electron transport chain that is present in the intermembrane space of mitochondria, and here it regenerates the NAD plus and also helps in synthesizing the ATP molecule. So let's begin with this glycerol 3-phosphate shuttle. Okay. So here what happens, this glycerol 3-phosphate shuttle, this is mostly seen in the skeletal cell of the body. Okay, this is mostly predominantly found in the skeletal muscle cells, skeletal muscle cell of the body. So what happens in glycerol 3-phosphate? See, there are this glucose to pyruvate, and this glycolysis process takes place in the cytoplasm. That means the produced NADH is present in the cytoplasm. Okay, that means NADH is present in the cytoplasm. So there is an enzyme. Okay, there is an enzyme that is known as glycerol 3-phosphate dehydrogenase. Okay. So here what this enzyme does, there is an enzyme that is known as glycerol 3-phosphate dehydrogenase. Phosphate dehydrogenase. So what this enzyme does, this enzyme will convert the dihydroxyacetone phosphate. This enzyme will convert the dihydroxyacetone phosphate to glycerol 3-phosphate. Okay, to glycerol 3-phosphate. Okay, and in this process, it also accepts NADH with a proton molecule, and here it produces NAD plus. It regenerates NAD plus. So what happens? This enzyme, the enzyme 1, that is glycerol 3-phosphate dehydrogenase, first convert the dihydroxyacetone phosphate, oxidize the dihydroxyacetone phosphate to glycerol 3-phosphate. Now this glycerol 3-phosphate will enter into the mitochondria. Okay, it will enter into the mitochondria. And here in mitochondria, in the inner membrane of mitochondria, in electron transport chain we have studied there are 4 complexes: complex 1, complex 2, complex 3 and complex 4. Okay, so here what happens, this glycerol 3-phosphate dehydrogenase is present at this point. Okay, this is the place. So this is the place of glycerol 3-phosphate. Okay, this is the place of glycerol 3-phosphate. So what happens here, this glycerol 3-phosphate will remove the electron and proton, 2 electron and 2 proton from the glycerol 3-phosphate. This glycerol 3-phosphate dehydrogenase again remove 2 electron and 2 proton from glycerol 3-phosphate, and here there is FAD is present. Okay, in the inner membrane of the mitochondria, FAD is present. This FAD, this glycerol 3-phosphate dehydrogenase will place this electron and proton to the FAD. Okay, onto the FAD, and FAD will convert to FADH2. Okay, and then this glycerol 3-phosphate will convert to dihydroxyacetone phosphate. Okay, after removing 2 electron and 2 protons, it converts to dihydroxyacetone phosphate, and this 2 electron and 2 proton will be accepted by FAD, that converts to FADH2. Okay, and here what happens, as this is present at the complex 2, between the complex 2 and complex 3 of the electron transport chain, so here this NADH will give the electron to the electron transport chain at the complex 3. Okay, and in complex 3 here it will pump out 2 protons. In complex 4 it will pump out 4 protons. Okay, the electron from the NADH, that is driven by the glycerol 3-phosphate dehydrogenase. Okay, now for one NADH, 6 protons are getting pumped up. Okay, as we have discussed earlier, 4 protons makes 1 ATP. That means 6 proton will make 6 by 4, that means 1.5 ATPs. Okay, so this is the glycerol 3-phosphate shuttle. What happens? This shuttle is predominantly found in the skeletal muscle cells of our body, and here what it does, there is an enzyme that is glycerol 3-phosphate dehydrogenase. This enzyme will oxidize the dihydroxyacetone phosphate to the glycerol 3-phosphate. Okay, that way for this glycerol 3-phosphate, this is known as glycerol 3-phosphate shuttle. Okay, on the name of this glycerol 3-phosphate. Now it can oxidize the dihydroxyacetone phosphate to glycerol 3-phosphate, and in the process it always also utilize the NADH and it regenerate NAD plus. Now the glycerol 3-phosphate will enter into the mitochondria. Okay, and here in mitochondria, in the inner membrane of mitochondria, in between the complex 2 and complex 3, there is FAD is present. So this glycerol 3-phosphate dehydrogenase will take the electron and proton from the glycerol 3-phosphate and give it to the FAD. Then FAD becomes FADH2 and the glycerol 3-phosphate will become again convert back to the dihydroxyacetone phosphate. Okay, this is the cycle that takes place in glycerol 3-phosphate shuttle. Okay, this is how the NADH moves through glycerol in the form of glycerol 3-phosphate to the mitochondria and into the electron transport chain to regenerate the NAD+. Now, let's move on to the second type, that is the Malate aspartate shuttle. Okay, so let's go for the second type. So as glycerol 3-phosphate takes place in the skeletal muscle cells, but there are some other cells in our body which does not support the glycerol 3-phosphate. They causes different type of shuttle system. Okay, these are Malate aspartate shuttle. So now we'll be discussing the Malate aspartate shuttle. So this Malate aspartate shuttle, these are predominantly found in the cardiac cells, cardiac muscle cells or cardiac cells, whatever you say. Okay, cardiac muscle cells and liver cells. Okay, these are predominantly found in the cardiac muscle cells and liver cells. So here what happens, as I said, glycolysis takes place in the cytosol, NADH is produced in the cytosol, but NADH has to move into the mitochondrial matrix. So here what happens, NADH has to take some of the form of some other products to go into the mitochondrial matrix, as it can't directly enter to the mitochondrial matrix. So what will happen here in Malate aspartate shuttle, this NADH moves into the mitochondrial matrix in the form of malate. How? So what happens, there is a lot of oxaloacetic acid present in the cytoplasm of the cell. Okay, there is a lot of oxaloacetic acid. Now this oxaloacetic acid will convert, okay, before that, this Malate aspartate shuttle has two key components, okay, two key antiporters. It has two key antiporters. Okay, these are malate alpha ketoglutarate shuttle antiporter, and the second one is glutamate aspartate antiporter. Okay, glutamate aspartate antiporter. Now what is antiporter? What is this antiporter? Antiporter is a specific type of transporter which transports solutes, okay, two solutes simultaneously but in different directions. That means one solute must be while one solute is moving in, at the same time another solute is moving out of the thing or out of the cell. Okay, so antiporter, what it does, it transport two solutes in the opposite directions. One solute is moving in while another solute is moving out. That is the basic function of antiporter. So here what happens, Malate aspartate shuttle, it has two main antiporters, two transporter system to transport the NADH. One is malate alpha ketoglutarate antiporter and the second one is glutamate aspartate antiporter. Okay, so here from this malate and aspartate, it is known as Malate aspartate shuttle. Okay, so let's begin with the mechanism. As I said, there is a lot of oxaloacetic acid is present in the cytosol of the cell. Okay, there is a lot of oxaloacetic is present in the cytosol. So now what will happen, there is an enzyme that is known as malate dehydrogenase. I am writing the enzyme names here, malate dehydrogenase. Okay, so oxaloacetate will convert to malate, okay, with the help of this enzyme that is malate dehydrogenase. Okay, malate dehydrogenase converts this oxaloacetate to the malate, and here NADH converts to NAD plus. That means this malate dehydrogenase takes NADH and oxaloacetic acid and then it produces the malate. Okay, now once the malate is produced, malate moves into the mitochondria. Okay, once the malate is produced, it is permeable. The mitochondrial inner membrane of mitochondria is permeable to the malate. So what happens? It moves to the mitochondria. Now what happens, once malate is entered into the mitochondria, it will release that NADH. Okay, and it will again revert back. It will again revert back to the oxaloacetate. Okay, the malate will again revert back to the oxaloacetate. And in this reaction, also what it does, it uses up some NAD to produce NADH. Okay, so with the help of this malate, NADH entered into the mitochondria. Okay, and here it used some NAD plus to convert back to the oxaloacetate in the presence of enzyme, again the malate dehydrogenase. So it is a reversible reaction that is catalyzed by malate dehydrogenase. Okay, it can this malate dehydrogenase can catalyze both the forward and the reverse reaction. Now what happens, there is other molecule is present in this malate alpha ketoglutarate antiporter, that is alpha ketoglutarate. So what is alpha ketoglutarate? As this alpha ketoglutarate acts as the transporter. Okay, so it has again it has to go back to the cytosol. Okay, for further process. So again this alpha ketoglutarate will convert. It's also reversible reaction that converts to glutamate. Okay, this alpha ketoglutarate can penetrate into the inner membrane but can go out, so it converts to a glutamate. Okay, and again the oxaloacetate that is produced here from the malate, this oxaloacetate will convert to aspartate. Okay, it is converted to aspartate in the process of transamination. Okay, in the process of transamination, amino acid transamination. That means amino acid transfer takes place. Okay, so what happens in malate alpha ketoglutarate? What it does, first the malate dehydrogenase combine the oxaloacetate that is already present in the cytosol with the NADH, then produce malate. Malate enters into the mitochondria. After entering into the mitochondria, it releases that NADH, and here this malate again converts back to oxaloacetate. Okay, and here what happens is oxaloacetate will again convert to aspartate to come out of the cell, out of the mitochondrial membrane. So oxaloacetate converts to aspartate, and the alpha ketoglutarate that acted as a transporter here, that alpha ketoglutarate will convert to glutamate. Okay. So now this glutamate and aspartate that act as an antiporter, that is glutamate aspartate antiporter, and that those glutamate aspartate antiporter again bring this NAD plus to the glycolysis process, or to the cytosol for the glycolysis process. Now this NADH that enters into the mitochondrial matrix it not only helps in the regeneration of NAD+, it also helps in the ATP synthesis process as it moves along the electron transport chain. Okay, so these are the two systems for the transportation of NADH from the cytosol, where it produced, to the mitochondrial membrane where it actually works. Okay, so today we have discussed the two shuttle system of for the aerobic cellular respiration, that is glycerol 3-phosphate shuttle and malate aspartate shuttle. Glycerol 3-phosphate shuttle, dihydroxyacetone phosphate with the help of glycerol 3-phosphate dehydrogenase converts to glycerol 3-phosphate. Here it takes that NADH. Glycerol 3-phosphate enters to the mitochondria. In mitochondria, it removes that electron and protein, and then this electron and protein is accepted by the FAD, and again it converts back to the dihydroxyacetone phosphate. Okay, that is glycerol 3-phosphate, and glycerol 3-phosphate predominantly takes place in the skeletal muscle cells, while cardiac muscle cells and liver cells use the malate aspartate shuttle for the transportation of NADH. So here what happens, oxaloacetate that is abundantly present in the cytosol, they are combined with NADH with the help of malate dehydrogenase, they converts to malate. Okay, malate enters to the mitochondria, and after entering into the mitochondria, it removes that NADH and it converts to oxaloacetate. Now this oxaloacetate will convert to aspartate. Okay, for the to move out of the mitochondrial matrix. Now this alpha ketoglutarate, the antipoda of this malate aspartate shuttle, this alpha ketoglutarate will convert to glutamate. Okay, now this glutamate and aspartate, they act as an antipoda, and again they move back, they go back to the cytosol. So that is the whole shuttle system, two types of shuttle system. So if you have any doubt regarding today's discussion, please comment in the comment box. If you like my video, please give a big fat thumbs up and share my video. Don't forget to subscribe my channel. Thank you so much.